Thank you. I'm Steve Kautz. I'm in the College of Health Professions, and I direct several NIH-funded centers, including relevant to this, the National Center of Neuromodulation for Rehabilitation. And in addition, while not being a zebra myself, I am married to one and have two children who are zebras. My daughter, who's severely disabled from it, and as a zebra amongst zebras, my son, who has EDS also, but is not as significantly affected. And so most of my career has been spent doing neurorehabilitation research in the stroke population and other populations like that. But of course, over the last 10 years, I've become very invested in the idea of seeing how little there is for EDSers in terms of innovative treatments and things like that. So when given the opportunity to start researching this, I've been thrilled to take our first steps in that. So I'm going to speak to you about a pilot study that we've done here at MUSC in looking at a non-invasive neuromodulation approach to EDS. Next slide. Oh wait, I have a number of co-investigators and great research staff that are here, and you will also hear from one of our investigators right after me giving some further results related to brain imaging of the study. So next slide. Oh, I'm sorry, the green button? Okay. So you've seen everything here today, and the movie couldn't have done a better job of pointing out the need for this type of innovative interventions for EDS. Worked so well, that first click. There we go. And you've seen this picture also, and this is sort of where the inspiration came from for this study, is that there are all these symptoms, but one of the things that you can note is that the vagus nerve has afferent and efferent connections with multiple of these systems, and there were studies in other populations that showed that vagus nerve stimulation had a positive effect in 9 of the different symptoms, the ones with asterisks there, in other diseases like functional GI diseases and POTS and some of these other populations. And so the thought was, well, maybe the autonomic nervous system is a big connector of what's going on here, and if we were to address that and try to do that we could have this effect. So I'll be talking, I've said vagus nerve stimulation, but we're actually doing what's called transauricular neurostimulation, because the device we use has 2 active electrodes, one of which stimulates the trigeminal nerve as well as one that stimulates the vagus nerve. And so what's great about this is that it's a non-invasive, safe approach, and it can be administered safely at home. As you know, it can be difficult for those with EDS to leave home, to come in for treatments. And to be able to do this safely in their home, even if they're bedbound, is a great positive. So our hypothesis was that stimulation of the autonomic nervous system could renormalize autonomic nervous system function and therefore directly reduce numerous symptoms associated with multiple different physiological systems simultaneously. And then if we are able to renormalize some of that function, then the system relief may persist over time. And so you've heard about, it's called a connective tissue disease, but maybe it should be called a neurogastroimmunological disease. You've heard, over and over from this population, and the vagus nerve is involved in all 3 of those systems. So this is our design. We had a 4-week study. The first 2 weeks, it was a double-blinded study where we don't know what we were giving them. They don't know whether they were getting active or sham for 2 weeks. And then after that, everybody got 2 weeks of open label where everybody was getting active. We didn't want people to end up with just a sham treatment in this, and yet we needed the sham treatment. So this design where we had the open label phase would allow us to do that. We were able to do some mechanistic measures pre and post the 4 weeks, and that was, and then we took a bunch of systems across these different symptoms across these different systems to look at. And okay. All right. So first of all, I'm honest, I'm not doing anything. There we go. Okay, it's done now. First of all, it was safe, and we've had more than 1,000 sessions now with no adverse events. 94% of the participants could properly and safely administer the wearable device without supervision after their second treatment session. So therefore, our study has suggested that it's a safe and feasible at-home treatment. A lot of words on here. What I do want to say is, so everybody in this study got at least 2 weeks. So if you just, because it's not a large study yet, if you pool all of that together to just see, is there an effect to this, what we found is that there was a very strongly significant effect on GI issues, the gastric symptom relief scale, musculoskeletal pain, head pain, were all very strongly significantly improved compared to baseline. In anxiety and dysautonomia symptoms were strongly significant in the relationship, and fatigue and vagal efficiency, which is sort of a physiological measure of autonomic nervous system function, were significantly related. Quality of life was almost, the depression was almost, and one of the immunological markers, interleukin-10, which is when increased as a marker of good immune system function, were almost significant here. So, and then we had a 12-week follow-up in 8 of the people, and for those, we found that there was a trend towards durability. Still not statistically significant, but there was trending that way, and this is 12 weeks after they stopped receiving stimulation. So that's all very encouraging. So we're excited about the possibilities, and I'm in the process of writing 2 grants right now to get further funding to do a larger scale study of this, to try to get this out there into the community as something that's FDA approved and insurance covered and available to people. So next slide. Again, we had some preliminary evidence of just now when we look at 4 weeks, we don't have as much data, so we didn't have as many of the things being all the way strongly significant, but there were some that were like anxiety and quality of life, when you only looked at the people who got 4 weeks of that, were strongly significant. And so perhaps there's this evidence that there is more effects of 4 weeks and that the 2 weeks wasn't enough. On the next slide. Yes, so we have some evidence of modest effects of 2 weeks when we look at the sample that only had the 2 weeks, but it does suggest that maybe that's not enough. So the grants that I'm currently writing are to look at 4 weeks as the minimum dose against 8 weeks with the same kind of a design, with a blinded 4-week phase and then an open label 4-week phase, to look into that, and we'll do some very good physiological measurements of the neural system, of the GI system, and of the immune system to really dive into the different effects that are out there. Next slide. So we talked about it as a hypothesis. Maybe it's the autonomic nervous system function that's impaired. So this is as often happens in science, I don't know why it is, but often the first subject can sometimes be a perfect example of just what you thought you were going to see, and you see that first date and you go, oh my gosh, you know, we've nailed it. This is going to be there. So this on on my right, your left here, the baseline. So you can see this test for vagal efficiency. They lay down for 2 minutes, they sit for 2 minutes, and then they stand for 2 minutes. And that those of you with POTS will know that your nervous system needs to adjust differently to each of those. And in a healthy person, you see something which is like the one to the far left, which suggests that there's this separation of each of those. You're looking at sort of the heart period, and so that needs to be separated because your heart rate goes up as you need to do more work with this. And you can see on the left that there's this very nice separation. But you can see in this person, when they started, before they received any stimulation, it was sort of overlapping and no real trend like that. So suggesting that there was abnormal autonomic regulation, potentially leading to some of these symptoms across these different systems. So overall, we have evidence for about a 50% increase in the vagal efficiency. It's barely significant at this point with what we've had, but we're pretty excited that this could be a mechanism for what we're seeing, and it really gives us sort of this physiological explanation of why this might work and why it's promising. Next slide. And you've heard a lot from we're talking about GI and talking about pain and talking about immune function. And, so it's been hypothesized that mast cell activation in the gut results in sensitization of the vagal sensory nerves that causes changes in the amygdala in the brain that are associated with the suppression of descending inhibitory pathways that take the nociceptive signals, the pain signals transmitted by the gastric spinal sensory nerves, with the combined mechanisms contributing to the pain and potential neuropsychiatric symptoms. So thus therapeutic targets could include normalizing any part of that loop where you have this autonomic, immune, pain interplay here. And Dr. Maitland has talked much about the sort of the close relationship between the immune system and the somatosensory system and all of these different parts. And so it's really a way that this neuroimmune interactome could be a part of what's going on. And so that's what we're doing now, is working towards that kind of research. And so I'm very excited to be able to present this, and I really hope that we're able to get additional funding to take this to the next level and be able to do this study. So any questions?
Could you share with us the dosing regimen? Are patients adapting to different levels of pain? How does that work?
They do 2 hours of stimulation a day for the period there. And so they do essentially morning and evening. They get an hour of stimulation is the dosing that we've done in this study. Mm-hmm. Yes?
So I was part of your study, and it really improved my balance during the study, and I actually have still been doing it for an hour a day ever since. Yeah, so I was part of your study, and I've been continuing to do vagus nerve stimulation for an hour a day. It really impacted my balance, as my physical therapist even saw while we were doing it. My standing balance, it was really incredible, and also kind of changed some of my autism experiences. And I've continued to use it we even went to Germany to get Neurostim, which isn't available in the United States yet, and is a little bit more similar to what you were doing for stimulation. So thank you for doing the study, because it's been really meaningful for me.
Oh, that's exciting. Thank you for sharing that.
Was there another question here? Oh, yeah, just a small one.
I was just curious, For the like the ear clips, I guess, for it, was it on just one or both ears?
On one ear in this study.
Okay, gotcha.
Yes.
I think you mentioned it hit both the vagus nerve and the trigeminal nerve.
Yes.
Do you have an opinion as to, are they equally important, one more than the other? Would you continue to try to hit both?
I think that the vagus is certainly important, and I wouldn't not hit the vagus there. The trigeminal, we don't have as much evidence for. There's some good evidence for it in pain populations, and this device had been used in opiate withdrawal as a way in what we'd used before, what had been used previously. Okay. I'll introduce my colleague, Xiaolong Peng, who is presenting the brain imaging expert, and he will be presenting results on brain imaging that we did pre and post in the study.